Hello everyone, good afternoon. I hope you've been having a wonderful time at the Crop Protection Forum. I'm sorry that I can't be with you there all today, but this last week prior to my departure from Australia has gotten quite busy. So I hope you all understand, but I'm glad to be able to have the chance to connect with you to talk about mapping weed genomes and what opportunities that may have for weed management. And knowing that I'm the last thing between you and the sundowner for the day, let's get into it. I want to show you today that we are well and truly into the era of weed genomics, with exciting possibilities opening new research avenues. We have from the International Weed Genomics Consortium a new review paper that's in review, but you can find the preprint online at bioRxiv as indicated here. And this outlines the various research questions that weed scientists around the world have come together to identify as their priorities using weed genomics. Chief among these, of course, are herbicide resistance, as well as understanding weed evolution, weed biology, seed production, aspects of non-target site resistance, gene flow, and stress adaptation. The International Weed Genomics Consortium is the result of a collaboration among global scientists since 2016 at the International Weed Science Congress in Prague. Out of a workshop there, some clear priorities were identified, including that everyone was finding that it was difficult to find the resources and justification to sequence a single weed genome, and then to also do the interesting science that would follow. So out of the idea of the consortium was the thought that industry partners would have an interest in obtaining genomes, and then these could be available for everyone, with the incentive for industry that they could have the genomes first for an initial confidentiality period. You can see that there was a meeting for the consortium in Rothamsted in March 2017, attended and included by Jenna Malone from Adelaide. And out of those discussions, a further workshop was held in 2017 at the Global Herbicide Resistance Challenge, followed by some additional meetings, a lot of discussions with lawyers and universities and companies and agreements, leading to the official launch of the IWGC in April 2021 as a 3-year project, which will be in its first phase finishing next year in 2024, and we're looking to extend into a phase 2. The IWGC is a public-private partnership that represents a community of scientists from academia and industry, with everyone interested in the genomics of weedy species. The vision is to obtain the reference genomes for the most important weed species worldwide, whether that's through the consortium or assisting with partners at other places or simply finding those genomes once they're completed. The second goal is to have a user-friendly online genome analysis tools and database, as well as training. And third is to facilitate discussions and collaborations within weed genomics. This allows us to find new ways to utilize genomics to improve weed management, our understanding of weeds, and also to bring in new scientists to work on this exciting area. The companies that were the founding members for the IWGC include BASF, Bayer Crop Science, Syngenta, and Corteva AgriScience, with the sequencing for weed genomes conducted at Corteva's genomics facility in Iowa in the US. The IWGC also has critical support from the Global Herbicide Resistance Action Committee. We've also received a major matching funding grant from the Foundation for Food and Agriculture Research in the US, which allowed us to greatly expand our training efforts as well as sequencing for more genome species. And finally, the USDA NIFA sponsored a conference grant for an IWGC conference that was held earlier this year. It's been a busy year for the IWGC in 2023. Including on the lower left, a symposium and training workshop held at the WSSA in January that was well attended, including by Australian scientists, and included opportunities for people to get hands-on with bioinformatics training, as well as hear some of the latest updates on weed genomics research. In particular, I think it's impressive the number of people involved here when we can consider some of the attendance at weed genomics sessions, say in the last 10 to 15 years. You can see there's clearly a growing community of people working in this area and working ahead with research to potentially improve weed management options into the future. On the lower right, you can see an EWRS training workshop that was held in March that was longer, more intensive for 3 days, and this format seemed to work quite well for people to really learn some of the bioinformatics approaches and applications. Finally, in the upper right, you can see a meeting that was held in September here in Perth for the PAG Australia and a weed genomics workshop that was held at UWA. The first two goals of the IWGC are to sequence and annotate the reference genomes for major weed species. Towards that goal, we're on the trajectory to have 51 weed species with at least one reference in Weedpedia, which is the online database, by mid-2024, which is quite exciting. As well, several species have multiple assemblies that have been completed, allowing something called pangenomics, which is where you can compare the genetic diversity in species across the whole genome assembly. Currently, there are 325 users registered in Weedpedia, which is an excellent growth over these last 3 years. We have 39 weed species that have structural and functional annotations in the database, meaning that their genes have been mapped out and we have some sense or prediction of what their function is, what those genes do. 19 of these species have been completed by the IWGC, and 22 have been completed by collaborating groups around the world. So we have about 12 more to get annotated and uploaded to the database. Of critical importance for Australia, of course, is annual ryegrass, Lolium rigidum. And a very excellent assembly was completed for ryegrass by CSIRO last year, and the IWGC also at the same time had an assembly in progress, and that's been completed by Corteva and the IWGC. Here you can see some high-level summaries of it, including the 7 chromosomes. 2 different haplotypes have been assembled. So as you know, ryegrass is a diploid. We've assembled the DNA sequence of both strands in this individual, with the exception of this small spot that you can see here on chromosome 5, where perhaps it's collapsed into one haplotype, but we are aware of that, and in general, this essentially gives us 2 genomes as well as the CSIRO, which gives us 3 ryegrass genomes for people to work with in their studies. Here you can see the alignments of the chromosomes to each other, and essentially this is an excellent assembly. It's what we call chromosome level, telomere to telomere, and this maps out all of the genes in ryegrass, which opens then the avenues for future work and better understanding. To summarize the state of weed genomics to this point in time, a lot has happened. Genomes have been assembled. Many people are working on this, a lot of enthusiasm in the research field to apply genomics to weed science questions. What can you watch for in 2024 and beyond? Well, the genomics database and analysis will continue to grow. You can check out weedgenomics.org, where you can sign up for the email newsletter as well as register for an account to Weedpedia, the online database, where all of this information is held. The assemblies and annotations are also going to be uploaded to NCPI, the public gene database, once those are completed and submitted for publication. So as well, you'll be able to download the information there. However, we do have the one-stop shop of the Weedpedia database to make it easier to access all of this information. In the phase 2, we're going to be working on improved annotation for just a few species, 3 to 4, ryegrass being 1. So that means we're going to dig deeper into the predictions of what these genes do, particularly the genes related to herbicide metabolism, non-target site resistance, as well as target site resistance genes, to make sure we have everything identified in the genome. As well, there will be more training and networking, so you can look for online videos that people could work on at their own time to learn bioinformatics approaches, as well as in-person workshops, and continue to have conferences, webinars, and communication around weed genomics. Next, I'd like to talk about how can weed genomics potentially change weed management, and give you 2 examples. One is around improving diagnostics for resistance, for species identification, and a second example, looking ahead to the future, what might we see for spray-on gene silencing technologies? Applying weed genomics to research questions in weed science will allow us to track things like gene flow of resistance, movement of species at the landscape level and regional areas around the world. This example shown here is for Palmer amaranth, a major weed that is native to the US and is a big issue for glyphosate resistance due to extra copies of the target gene, EPSPS. And that target gene is carried on a loop of DNA called an extrachromosomal circular DNA. It's in a way kind of an extra chromosome that's stably inherited. In the study that was led by Paul Neve at Rothamsted Research in a collaboration with South American researchers and our group at Colorado State, we got samples of Palmer amaranth from around the US, both glyphosate resistant and sensitive, as well as from Brazil, Argentina, and Uruguay, where the species has been introduced. using genome-wide molecular markers, as well as ways to measure the eccDNA, we were able to show that there were a couple of different independent introductions. Certainly Argentina and Brazil have had separate introductions, and possibly Uruguay had its own introduction. We could also find that glyphosate-resistant populations in Brazil and Uruguay carry the eccDNA found in the US. So this information can be used to help inform things like regulatory quarantine requirements, monitoring, as well as new species identification, as Palmer amaranth in particular continues to move around the world. Here's the second example of using genomics to track the evolution of resistance and movement of a species. This is for kochia, a tumbleweed found in North America. And so for myself, I'm located here in the northern part of Colorado. What we did in this study was, by assembling the kochia genome and then further assembling the locus, the spot in the genome where the EPSPS gene has been duplicated, we were able to determine that there was an original genotype of this gene duplication that occurred here in the central Great Plains of the US, shown by these red plus signs. We also found kochia from the northern part of the Great Plains, as well as the Pacific Northwest in the US, that have extra copies of EPSPS. However, their genotype for how this happened is different. They don't have the same markers. And that leads us to conclude that resistance has occurred a couple of different times, but as well, we can track the movement of low frequencies of these genotypes into the other regions, telling us that individuals of this species are able to move across the continent, across very large regions, and by understanding the epidemiology of that movement, gives us potential to intercede and hopefully interrupt by showing that this movement at the very large level is possible, as well as potential for independent occurrence of glyphosate resistance in the first place. Now, big picture, where can weed genomics take us to improve weed management? One way that this could go, and so this is just kind of an idea for everyone to think about, if we compare to medicine, where people are thinking that the current medicine is essentially one treatment fits all. If you have us present with a certain disease, you get a certain therapy that might work well for some, okay for others, and maybe even be negative for some subset of patients. But the future, they're looking at, how can you not only have the information on the disease, but look at your genotype, your genome, your blood markers, etc., other types of information, to then have a personalized therapy that would work better for everyone, of different backgrounds, different types of individuals. So instead of thinking about medicine, what about weed management? In some ways, we might tend to apply the same management practice, same herbicides to a field. We do have some information on that, right? We can do herbicide testing, know what mixtures or modes of action would or wouldn't work in a given situation. But could we really amplify that and have much more information, moving towards a biobank for weeds? For example, in ryegrass, could we start to build a database where we have the whole genome resequencing of known resistance phenotypes? That builds up then a database where we can both characterize known resistance mechanisms and use modeling, artificial intelligence, etc., to try to deduce currently unknown resistance mechanisms. We know there are a lot of them out there. Then over time, we can build up with more rapid sampling. Someone could have their ryegrass samples genotyped for these known and characterized resistance mutations, further building the information database and using this as another piece of information to guide herbicide recommendations. In particular, this may be useful for those modes of action and herbicides where resistance is maybe newly emerging. If we think about the importance of pre-emergence herbicides like pyroxasulfone, prosulfocarb, sinmethylin, trifluralin, etc., for ryegrass management, but where resistance is perhaps initially occurring or not as widespread, or maybe there's an opportunity for intervention, now would be the time to have this rapid information and move towards a personalized weed management in which the right treatment for each management unit can be correctly applied. All of this thinking about the power and potential of weed genomics can give exciting ideas for new innovations in weed science. One that I think we can often talk about, it's an interesting example, is rapid tests, rapid in-field diagnostics. On the left, what you're looking at is from blackgrass, as you've heard about today. There's a gene that encodes a protein called GST-F1 that's a great marker for resistance. And what you're looking at here is a rapid flow cell test with an antibody in which you have a test strip and a test bar, and then if the control line also lights up, that means that the protein is present, and likely that sample does have multiple herbicide resistance. We'd have a lot of work to get to make this happen for all the various resistance mechanisms are out there, but it is a great example of where things can go. I'll talk a bit now about RNA targeting, spray-on gene silencing, but can we use this both as a way to reverse current herbicide resistance and perhaps as a new herbicide on its own? And finally, just thinking of some of these ideas, could we actually use traits from weeds like stress tolerance, disease resistance, as then sources of new genetic variation to improve crops? I don't have time to go into all the details of RNAi and antisense pathways for gene silencing, but if you're interested, there is a review paper, recent one here in Advances in Weed Science, that summarizes very well the challenges and opportunities for spray-on gene silencing, particularly with the view of RNAi, but it's applicable for the various pathways. One of the key areas that's needed to make it work is weed genomics, and so that is really getting covered in a good way for species. Of course, more work to do, but that's on its way. The rest of the research is around formulation and delivery, stability, how to actually make this work, and then to find the right targets, whether that's reversing herbicide resistance or as new targets on their own. But there is a lot of opportunity and potential in this research area. Some of the specific companies are startups that you might keep an eye on working for spray-on gene silencing. You could think of this as nucleic acid in a jug, in a barrel, so to speak, that this would be a product like a herbicide, but instead of targeting proteins, you're targeting RNA. This is a great partner potentially for precision spraying applications, both due to the cost and to just help make the whole system more precise. You could think of these as next-gen pesticides that also have the potential for design-based resistance management and response. So not that these are any less prone to resistance, but that there's the opportunity to monitor mutations and potentially adjust the spray-on gene silencing targets over time. Here I've shown a few companies I know that are working in RNAi, including RNaissance, Nanosur, Trillium Ag, and then on the right I'm just highlighting that I'm working at CSU with an antisense platform called Ohm Silence, and this is our partner Vinu Aishwarya with Ohm LifeTech. So we're using the antisense pathway and in early stages of research to address the delivery to use that kind of gene silencing as potentially a new herbicide. With that, I'd like to end and say thank you for your attention and remind you that you can connect with the International Weed Genomics Consortium at weedgenomics.org. I have my email and Twitter listed there. I also like to especially thank AHRI and UWA as my hosts for sabbatical this year. It's been a wonderful experience, as well as my collaborators at the University of Adelaide, who I've worked with a lot over the years, and other collaborators across Australia. I'm pointing out the funding for IWGC and FR, and as well as my collaborators to the IWGC and back at CSU. Thanks very much, and have an excellent evening enjoying what's been a great conference today.